Today on G Fun Facts Online, we're diving deep into the microscopic universe within our own cells, thanks to a mind-blowing technology called cryo-electron tomography, or cryo-ET for short. Imagine being able to see the tiny, intricate machinery inside a living cell, not just as a flat image, but as a stunningly detailed 3D model. And when I say detailed, I mean so detailed you're getting close to seeing individual atoms. This isn't science fiction, it's cryo-ET, and it's revolutionizing how we understand life itself by showing us cellular components in their natural active state. So how does this incredible technology actually work? It's a bit like creating a miniature frozen diorama of a cell, and then taking super detailed 3D scans. First, scientists take their cells, or tiny bits of tissue, and place them on a special grid. Then comes a crucial step called vitrification. The sample is plunged into liquid ethane, which is incredibly cold, freezing it almost instantly. We're talking milliseconds. This ultra-fast freezing is key because it turns the water in and around the cells into a glass-like state, not icy crystals. This means the cell's delicate structures are perfectly preserved, exactly as they were in life, fully hydrated and undamaged. Now, cells can be pretty thick, and for electrons to see inside, we need a thin slice. If the sample is too chunky, scientists use a technique called cryo-focused ion beam milling, or cryo-FIB. Think of it as a microscopic sculptor using a beam of ions to carefully shave away parts of the frozen sample, creating an ultra-thin, transparent window, typically only 80 to 300 nanometers thick. That's thousands of times thinner than a human hair. Once the sample is perfectly prepped, frozen and thinned, it's moved into a powerful transmission electron microscope, all while being kept at cryogenic temperatures. Inside the microscope, the sample is tilted at many different angles. At each angle, a beam of electrons passes through it, and a 2D image is captured. This collection of 2D snapshots from different perspectives is called a tilt series. Finally, powerful computers take all these 2D images and, using clever algorithms and a process called back projection, reconstruct them into a 3D volume. That's our tomogram. It's a detailed 3D map of the cell's interior. And if scientists want to zoom in even further on specific molecular machines, they can use a technique called subtomogram averaging. They find many copies of the same structure, align them, and average them together. This cancels out noise and can reveal structures at near atomic resolution. Why is cryo-ET such a massive deal for science? Well, the advantages are huge. Firstly, we get to see cellular structures in their near-native state, fully hydrated, unstained, and unfixed. It's like watching animals in their natural habitat versus a museum exhibit. Secondly, it provides true three-dimensional information, which is essential for understanding the complex spatial relationships inside a bustling cell. Thirdly, the resolution is just staggering, down to the nanometer and even sub-nanometer scale, allowing scientists to identify individual proteins by their shape right where they function. This leads to the fourth point, in situ structural biology. We study molecules inside the cell, in their crowded natural environment, which is far more revealing than studying them in isolation. And lastly, it's generally label-free, meaning no artificial tags or stains are needed that could potentially mess with what we're trying to observe. So what amazing things are scientists looking at with cryo-ET? The applications are incredibly diverse. They're visualizing the detailed 3D architecture of organelles, like mitochondria, the cell's power plants, or the endoplasmic reticulum, its manufacturing hub. They're mapping out the cell's internal scaffolding, the cytoskeleton, and determining the structures of massive molecular complexes like ribosomes, which build proteins, or nuclear pore complexes, the highly selective gateways to the cell's nucleus. Cryo-ET can even capture snapshots of dynamic cellular events. Think cell division, how cells engulf material, or how viruses assemble and infect host cells. This is critical for understanding how pathogens like viruses and bacteria interact with our cells at a molecular level, paving the way for new therapies. It's also fantastic for studying the intricate world of cell membranes. The field of cryo-ET is advancing at lightning speed. Sample preparation is getting better and more automated. New, faster detectors mean data can be collected much more quickly. Artificial intelligence and machine learning are revolutionizing image processing, helping to sharpen images and identify structures more efficiently. There is also cryo-correlative light and electron microscopy, or cryo-CLEM. This powerful combo uses light microscopy to find specific, fluorescently tagged events or structures of interest, and then cryo-ET zooms in for the ultra-high-resolution 3D details. Scientists are even pushing for time-resolved cryo-ET, aiming to capture cellular processes as they happen, like creating a 3D movie at the molecular level. And efforts are ongoing to image thicker samples and even entire tissues. Of course, such an advanced technique comes with its challenges. The electron beam can only penetrate so far, so very thick samples are still tricky. The raw 3D images can be noisy, making it hard to see small features. Biological samples are also very sensitive to radiation from the electron beam. Processing the enormous datasets requires significant computing power and expertise. And finding very specific, rare molecules or events within the vastness of a cell is still a hurdle, although cryo-CLEM is helping. Finally, while automation is improving, the overall workflow can be quite intensive. Despite these challenges, cryo-electron tomography is undeniably at the cutting edge of cellular imaging. It offers an unparalleled ability to visualize the 3D organization of cells in their natural state, down to near atomic detail. As this technology continues to evolve, becoming more powerful, automated, and accessible, it will undoubtedly continue to provide profound insights into the fundamental mechanisms of life, from molecules to whole cells, transforming our understanding of health and disease. It's truly a window into life itself. Thanks for joining us on Fun Facts Online.